Hello, and welcome to the next in the series of Bitesize Bioinformatics webinars looking at machine learning and examples in biological sciences. So this is the fourth in the series. There will be only one more after this. And what we're going to look at today is this paper here, looking at an approach for interpreting random forest models and its application to the biology of aging. So as I said previously, it's not so much like a journal club going through all the details of this paper, but just using it as an example to think about some of the concepts around the sort of machine learning models that we're using. So again, just another overview of what we'll cover. So we'll start off with a very quick introduction to machine learning itself, just in case anyone hasn't seen any of the others. I'll then introduce the paper quickly, look at some of the data sets they used and also the pre-processing that they've done. Then we'll go through the model that they used and how it works, sort of conceptually. And then we'll see how well it works, and potentially have a run through the model in tidy models. Although I'll see how we're doing for time at the end. I might just go through it in the slides and show you the code that we can use. So what is machine learning? In a normal data analysis workflow, it would look something like this. So you will collect some data, you will visualize and explore it, and probably also do some filtering and some cleaning, and sort of loop through this until you fully understand the data set that you have. Then you will usually formalize it with some kind of statistical test, which will give you some outcome that you can report. So whether or not you have a statistically significant difference between your groups, for example. When we're thinking about machine learning, we're still going to follow these same steps at the start, but instead of formalizing with statistics, we will formalize it through modeling our data. And we can use this as an outcome. So we can just show that we can create a model, and that might be what we're interested in. Or we might also want to use it for prediction, so that we can predict new data where we don't necessarily know the outcome. Today, we're sort of going to be looking at one where we are mostly reporting on our model itself, rather than looking in too much detail at the predictions and how good they are. So slightly different to previous ones. So again, the steps in machine learning that we might follow are firstly generating data with samples where the outcome that we want to predict is known. We will need potentially quite a large data set for this. And we obviously want it to be high quality so that we can make good predictions at the end. We can then pick a modeling method and build our model, and then evaluate our model and refine it to get the best model that we can from the data that we have. And then we can use it to make predictions where the outcome is not known, and then test the predictions that we have made. So how do we decide between different model types? The big things that we can think about are the actual data that we have and what we want to predict. So what kind of outcome type we want. So if we want to make quantitative predictions, we will need to use a regression model. So if we're trying to predict height, for example, we will need regression. If we want to predict a categorical prediction, like for example, if a sample is healthy or not, then we will need to use a classification model. Some model types can do both. Some can only do one or the other. And the other consideration is the input type of data that we have. So some models require all variables to be numeric. So we might need to do some pre-processing to meet these requirements. So for example, converting categorical values into numbers. And also some of them have expected behavior of our input data. So some expect it to be normally distributed, some don't mind at all. And there's also variation in the number of measures that we might want to use as inputs to our different model types. So some work pretty well with small amount of data, some don't scale very well to really large data, and some need a lot more data in order to work well, essentially. So different machine learning model types. We've gone through a few of these now. And today we're going to look at random forests. But in order to do that, we're also going to have a look at decision trees. So we're going to talk through both of these here. So without further ado, we'll get on to the paper itself. So as I said, we're looking at this paper, which is a new approach for interpreting random forest models and its application to the biology of aging. So as we might expect, it has to do with aging. The model that they're trying to build is looking at predicting genes with altered expression with age in the brain. So looking at genes that are overexpressed, underexpressed, or unchanged in the brain in young versus aging samples, essentially. So they got their gene expression data from GEO and AgeMap, which are just two big data sources, essentially. So from AgeMap, they combined cerebrum, hippocampus, and striatum expression datasets into a single dataset. So from this, they got 118 brain samples and associated 6,712 mouse genes. From GEO, they searched for gene expression and series datasets with expression levels in different ages or developmental stages. So they had, data across a few different time points. They then removed any unsuitable data sets, so things that had like single pathways or specific diseases or mutants and treatments. So they only used age-related data from healthy, adult, and non-treated samples. And only included data sets that had more than 2 adult time points as well. So from this GEO search, they ended up with 28 data sets. And within these contained 1,212 samples with different numbers of genes per dataset in these cases. So they then did some pre-processing, as we always do. So they filtered out genes with more than 30% missing gene expression data across all of the samples. And then they did some very basic imputation. So they just replaced missing values by the probe's average overall. They also searched for human homologs for mouse and rat protein coding genes. So they used only human genes as an input, although some of them were from other species as well. But to include these, they only included ones that had high confidence and that had like one-to-one orthologs. So they were pretty confident that they're equivalent, essentially. They then described the genes, which they also called instances in here, by features that were representing the presence or absence of a Go term. So using these genes, they decided if they were under or overexpressed in aging samples. And then they also pulled out whether or not they were present or absent in particular Go terms as a sort of binary feature. So just noughts or ones for all of that. Again, Go terms with less than 10 instances were removed. So if there were fewer than 10 genes associated with a Go term across the data set, that would be taken out. And so they ended up with a data set with around 17,500 genes and 7,500 Go terms, which are their features that are going to be used as predictors. Another thing to note with this is of the 3 classes that they split their genes into, there are quite different proportions in the number of genes within each of these. So the vast majority of them had no change of expression, Very, very few of them were underexpressed in age, and around 2.5% were overexpressed in age. So the model used, our input is essentially the presence or absence of genes in Go terms based on our different categories, whether or not they are over or underexpressed or unchanged with age. And we also want to pull out some kind of feature importance of the Go terms. So we will come back to this later, but essentially what they were interested in is whether we can pull out Go terms that are highly predictive of being over or underexpressed in age in the brain. So we need some kind of classification, and we have sort of numeric variables because they're noughts and ones, although in this case it doesn't matter because we are going to do a random forest. So in order to explain random forests, we're first going to have a look at decision trees. Decision trees look something like this. You have some kind of hierarchical system of questions that you answer in turn to decide whether, in this case, your sample is high or low. So to go through a quick example of how this works, this is just a sort of little data set where we're trying to predict cancer risk based on obesity, whether or not someone smokes, whether they exercise, and their age. And so we came up with this decision tree. If we have this unknown sample here, we can just follow through our decision tree. So do they smoke? In this case, yes. So we go down this side of our decision tree. Are they less than 50? They're 48 in this case. So yes. And do they exercise? No, they don't. So then we would classify them as high risk. Super simple, super obvious how this works once you have your decision tree. But how do you build the tree itself? So we have some kind of population or set of observations in our data. So how do we decide which variables to use? And also at what point do we use each variable? So here we had smoking at the top, but there's nothing really stopping us putting exercises at the top. How did we decide to do that? So we can calculate something called an impurity score and then pick the least impure variable to split the remaining data. And essentially this means that we can use the most cleanly predictive question to improve the tree. So we want to split our data into high and low as early as possible. So what does this actually look like and how does this work? So we're going to start off with 100 samples in our starting data. In this case, there are 30 high risk and 70 low risk. If we ask if they are smokers or not, we can then split that into yes or no and see what our numbers are of high risk versus low risk in each of these. We can see this outcome is impure because there is a mix of high and low risk individuals in each of these nodes. So yes, here is a node, no, here is a node, and they both have both high and low risk individuals within them. So what we can do is calculate how impure each of these nodes are by using a node impurity measure. And this can be based on essentially different probabilities. So to calculate our node impurity, we can do 1 minus the probability of a high risk individual minus the probability or the proportion, sorry, of the low risk individuals squared. So for the yes node over here, that would be 1 minus 18 over 20 squared minus 2 over 20 squared, which gives us a node impurity of 0.180. And we can do the exact same for our no and get a node impurity of 2.55. See, that's great, but really we want to know how good our question is rather than how good these nodes are. So in order to help us decide that, we can calculate a weighted average of our two node impurities here. So if we do 0.18 times 20 out of 100, which is the proportion of our original data that went into this yes node, plus 2.55 times by 80, again, the proportion that went into the no node, and calculate that, then we get a weighted average of our node impurities of 0.24. And that essentially describes how good our question is up here. We can repeat that then for each possible question or predictor, and then select the lowest impurity score at the end of it to get the best separation between our high and low at this particular point. A couple of additional things to notice or to mention is this node impurity is called a Gini impurity. We will come back to that in more detail later. And we can also calculate something called the Gini gain, which compares the weighted average of the new nodes with the impurity of the parent node. So what does that mean? If we also calculated a node impurity in this way for this smoker node, then that would equal 0.42. And if we do 0.42 minus 0.24, which is our weighted average of our new nodes, then we get a Gini gain of 0.818, sorry. Again, we will mention this again in a bit, but just to introduce those concepts while we're looking at this overall. The other thing we can do with trees is prune them. So sometimes lower branches don't really provide additional information, and we don't really need the bottoms of our trees or the bottom leaves to be completely pure. So it might be that we want to stop our trees early and just pick a majority answer. So if we carried on with our high versus low risk of cancer, it might be that at the end we have a question of whether they swim or not that can perfectly split our high and low risk. But maybe this doesn't really provide additional information. It's just going into a level of detail that's not really biologically relevant. So we might want to stop it at this point and just classify all of these as high risk. One way that we can determine at which point we stop is by setting a minimum node size. So it might be that once we have fewer than 10 of our samples in a node, we will just stop it there and say anything else is going to be too much detail for what we're really interested in. And so we'll simplify our tree and stop it earlier on. So that's decision trees. They are super cool, potentially, but they are also prone to overfitting. They are too specific to the training data quite often, and also deciding on particular factors like what point to stop can be sort of more tricky. They're also deterministic, so you get the same result every single time with a decision tree. The way that we can improve on this is by having many different decision trees rather than just one, and that's what random forests are. So random forests use something called bagging, which is a combination of bootstrapping and aggregating. So what actually are these? So bootstrapping introduces a couple of levels of randomization to our random forest. So the first level of randomization is by randomly subsetting our original data. So what we'll do is we will randomly select samples within our data without replacing them, or with replacing them every time. So you can select the same sample multiple times, essentially. So we randomly select them until we have a new data that is the same size as our original data, but will not include some samples and will include repeats of some others. So for example, this sample here that is age 32 and low, we can see has been repeated twice in our new random data, whereas the first one here and the last one here haven't been included at all. The ones that haven't been included, in this case these three, are called out of bag or OOB, which we will again come back to in a minute but is super important and another useful thing in random forests. So then, using our new randomized data, we will build a tree, but we will also add another level of randomization, which is that we will randomly select which variables we could possibly use at each branch point. And so what I mean by this is we will, for example, say that we can only choose from 2 different variables at each level. And so at the top level, we can only use smoker or only use exercisers. We just choose between those 2. We can't choose age or obese at this point. And then maybe at the next one, we can only choose between age and exercise, and then we can only choose between age or smoker. This just adds another sort of level of randomization within our random forest, which hopefully makes it more robust at the end as well. So then we will repeat this across hundreds of different trees. So we build a forest from our slightly randomized different decision trees. What we can do is evaluate our trees. So the out-of-bag data that wasn't included when we were building our decision trees can be run through them to see how well they predict those data they haven't seen before. So that's a really useful way of evaluating as you're building your model. And you can also optimize the random variable number and the number of trees that you have in your forest as well. So there's a couple of sort of tuning parameters that we can have a play around with at the same time. When we predict new data, we can run it down all of the trees rather than just the one, and then we count the predicted outcomes from each of them, and the most frequent outcome wins. So that's where we're sort of aggregating across all of our trees to get a hopefully more robust prediction based on however many trees we have included in our forest. The other thing that you can pull out here is it might be that, say, you have 100 trees and 80 of them predicted a new sample to be high and 20 predicted a new sample to be low. So that would mean that 80% of the trees predicted the same. And so therefore you might be more confident in that than, for example, if 50% of your trees predicted high and 50% predicted low. So another note on feature selection. When we're building these random forests, because we have so many different trees and different sort of randomized elements on where our variables can fall, we can also use this information to pull out more details about what is important within our predictors. So on the whole, more informative features or more informative predictors will appear higher up our trees because at each point we're trying to split our data so that it has the lowest impurity. And we can aggregate this information across our whole forest and pull out which ones generally appear higher up and therefore are likely more informative for predicting whether or not we are high or low risk in this case. So if we look across all of our forests, it might be that smoking comes really high in all of them, and maybe age is less important and comes more towards the bottom of the trees. And so therefore we can conclude that perhaps smoking is more informative and potentially more biologically relevant than age is for this particular instance. There are actually a fairly large number of different measures of feature importance that can be used to try and figure out which particular predictors are most important. And so I will go through a few of these at fairly high level sort of summarizations, just to give you an idea of some of the different approaches. So the first one here is the Gini variable importance measure. This looks at our Gini impurity scores and uses the out-of-bag samples to measure how much this decreases when using a particular feature of interest to split a node. So we're just looking at our impurity scores and seeing on new data how well that works. There's also something called the permutation variable importance measure, or VIM. And this looks at random permutations of all of the values of all of the observations for a particular variable of interest. So essentially, you will run a decision tree or a random forest, or one decision tree in your random forest. You will randomize the values of a particular predictor that you're interested in. So if there was any correlation between it initially, there would no longer be once you have randomized it. And then you look at how much worse the prediction is based on the randomization of the variable that you're interested in compared to the original data. And if it's a really good predictor, if you randomize it, you're going to end up with a much, much worse prediction accuracy based on your, again, out-of-bag data. For this, you do it for each tree individually and then average it across all of the trees in your forest. There is also a slightly updated version of this, which is called a conditional permutation VIM, which is essentially the same, but it corrects for some of the bias for correlated variables. So if some of your predictors are correlated with each other, it can cause some biases when you are just using this random permutation up here. The next one is a variable selection based on minimal distance. So this is essentially what I was describing previously. It calculates the average depth of the features in the random forest and assigns greater importance to features that are closer to the root or closer to the top across the random forest itself. There's also variable selection using random forests. This removes the least important features from a random forest until the predictive accuracy of your out-of-box measures is significantly reduced, and then uses the smallest set of features that have a predictive accuracy that's equivalent to the best random forest using all of the predictors. And finally, there's something called the intervention in predicted measures, which for each tree calculates the percentage of times that a variable is selected and then averages this across all the trees. And this calculates essentially the relative importance of each of these features based on its relative frequency for the use of classification. The choice of approach to this will vary depending on some different factors. Different approaches are more or less susceptible to different biases. So some of these assign more importance to categories that maybe have larger numbers of categories as an option, or maybe more influenced by the scale of the measurement of any particular variable. So if you have a variable that has really high values overall, it might be deemed more important than one that has really low values overall. And also sometimes the type of predictor, so some of them preferentially select numeric predictors versus categorical. They also, however, differ in the complexity and time required to calculate. So this sort of simplest one here, the Gini variable importance measure, has often been used but is prone to some biases. The permutation VIM is sort of considered probably a bit better, but also because you're doing these random permutations and things, it can take a bit longer to calculate and is maybe a bit more intensive. So if you're not really struggling with biases, you might want to go for a simpler approach. I mentioned these, because in the paper that we're talking about, they sort of came up with their own feature importance measure, which they called Compact Plus FV algorithm, which stands for computing the predictive accuracy of random tree rules with positive feature values. Essentially, the main headline is that it only looks at rules that contain positive values. In this case, positive values means that a gene is annotated with a particular Go term. And so therefore, this is sort of preferable than looking at everything because you have a sort of higher level of confidence associated with positive terms than with the negative terms. This comes back to the fact that positive ones are evidence for a particular Go term, whereas negative values are just a lack of evidence, which is not necessarily the same thing as there being actual evidence for it. Also, on the whole, it's more informative biologically. So if a gene has a particular biological property, it will have a positive value, as opposed to a gene just not having a property, which isn't necessarily as informative overall. In order to calculate this, it extracts two statistics for each feature and class over each tree. So it calculates something called the out-of-bag coverage, or OOB coverage, which is the number of genes that are passed through a particular rule containing a positive value to predict its class. And also out-of-bag hits, which is the number of genes that are correctly classified by rules containing the positive value. I will visualize this in a second, so if that doesn't make sense, don't worry too much. It will be easier to see once we see the little diagram. Again, we calculate this for each tree and average it across the forest, and we end up with an importance score for each feature. So each predictor and also each class, so whether they're overexpressed, underexpressed, or unchanged. And so we can look at the precision for each of the rules to predict a specific class. So what this means is that we can pull out essentially Go terms that are important for classifying expressed genes, Go terms that are important for classifying overexpressed genes, and also those that are important for classifying the genes that do not change. And we can pull those apart separately so that we can look at what each of the Go terms are for each of those classes. So I said we'd go through an example that will hopefully help to visualize this slightly more. So in this case, we're looking at this Go term here, which is for exocytosis. And we're trying to see or calculate the importance of this in relation to the no change of expression with age class. This is the example they include in the paper. So to calculate our importance measure, we are just going to do that for this here. So we can see this is the Go term we're interested in. We're only interested in the positive values for this. So we don't care about this one here because it's not associated with this particular Go term. We're only interested in the things downstream of our tree on the positive side of this. So to calculate our importance measure, we're first going to go this way and this way. So the first rule over here covers 36 out-of-bag instances. So there are 36 of our out-of-bag samples that came down this way to be predicted down here. And of those 36 that followed the tree this way, 35 of them were correctly classified. So 35 of the 36 that came down here were actually no change of expression. We can then do that for the second one, which is down here. So we went positive here and then negative for the next one and negative for the one after that to classify it as an N again. And in this case, we had 150 out-of-bag instances that came down this way, and 145 of them were correctly classified. And then we do that for the third one over here, where again we had 10 that came down here and 7 of them were correctly classified. Then we take all of these measures and combine them together to get a total rule-based precision. To do that, we add up all of the hits and divide them by all of the sort of instances that came through here. So we have 35 plus 145 plus 7 divided by 36 plus 150 plus 10, which gives us a total precision of 0.9541. And that gives us some kind of measure of how important this particular Go term is for predicting this particular class here. So, slight sidestep, just to talk about how they evaluated their model as well. And then we'll talk through some of the model parameters and then see the outputs based on this new feature importance sort of measure. So to evaluate their model, they used the area under the receiver operating characteristic curve, or the AU-ROC. The ROC curves plot the model's true positive rate as a function of its false positive rate. I'm sure many people will have seen these before, but they look something like this when you visualize them. If you had a perfect classifier, you would have just a sort of flat line on each, going all the way up to 1. If you have a random classifier, it would fall on the diagonal here. If you have a classifier that is worse than random, that's not great. So probably don't want to be using those. But often you'll see curves that look something like these ones here, with this one being the best classifier there. Again, they're calculated for each class in turn. So because we have 3 classes, it's not just binary. And then they take the weighted average. So again, accounting for the number of instances in each class across the 3. They actually use 10-fold cross-validation, which is where they split their data up into 10 sections, and then they will run a model on 9 out of 10 of those sort of sections across 10 different testing sets, so that each chunk of data trains 9 models and gets tested on 1 model. So that gives you an overall idea of how well your model is working. That is less influenced by just randomly which proportion of your data you happen to select. So that's how they evaluated it. What kind of parameters did they use? So they actually used, in this case, 2 different levels of cross-validation. So they used the overall 10-fold cross-validation, but within that, they use a 5-fold cross-validation to optimize parameters of the model at the same time. So for this, there were 2 main parameters that they optimized using this 5-fold cross-validation. Something called M-tree, which is the number of randomly sampled features for each step of the random tree, or the decision tree within the random forest. They tested 3 different values for this, which is the square root of the number of features times by 0.5 times by nothing, or times by 2. And they also optimized the number of trees in their random forest, looked at either 100, 200, or 300. And within these 5-fold cross-validations, they looked at all different pairwise comparisons. And then for their whole 10-fold cross-validation, they used the pair of these 2 values with the highest predictive accuracy, or the highest area under the ROC curve. They then used, for a final model, the values that were most frequently selected across all of the 10 cross-validation folds. So for each of the 10, they had potentially a different pair of values based on this 5-fold cross-validation. And then for the final model that they built, they used the ones that were used most frequently across the 10 different folds. And those values were an M-try value of 43 and a number of trees of 300. Another thing that they did when building their model was using something called undersampling. So at the start, I told you that they have quite large imbalances between their classes. So no change of expression accounted for 96.8% of the data, versus overexpressed, which was 2.4%, and underexpressed, which is 0.8%. The trouble with this is it can lead to models that are quite conservative when trying to predict the minority classes. So it will sort of by default just categorize more things as no changing just because they have there's such a large proportion of the data essentially. So they did something called undersampling, which is where they undersampled genes with the classes of either N or O within the in-bag set. So for the data that was actually used to train the random forest, they randomly deleted genes with class N or O so that they had the same number of genes as those in the U class. So it just meant that the data that was used to train the random forest was equal in numbers between the different classes there. And they found that this ended up with a better predictive accuracy than the random forest without undersampling. So you can see over here, we have our AUCs for each of our classes with undersampling and without. So it actually doesn't make a crazy amount of difference, but it is slightly better for each of them, and particularly, yeah, on the overall as well is better as well. Another benefit of this, so even though it hasn't necessarily improved these by a crazy amount, it has also massively sped up the running time. So because you're reducing the size of the training data, when using undersampling, each cross-validation run took 3.8 hours as opposed to 70.4 hours when you don't use this. So obviously a massive difference in the amount of time it took to train the model, which in itself is probably enough to justify this, particularly when it's also improving the accuracy of the model overall. So that's pretty much what they did with their model, and the sort of considerations behind it. So using their variable important measure, which is this compact plus FV, they compared it to the IPM score, which is sort of what it was based on, and looked at the Go terms that were identified by each of these. So this one here is the measure that they created in the paper. This is the previous one. They found that the IPM measure had similar features for all of the classes. So because most of the Go terms that were predictive in a sort of negative way, so as in they weren't found, these had an overall larger impact on this measure. And so a lot of the classes that this one found were sort of broader. So there are a couple of just neuron ones, which it's brain samples, so that's probably not going to be super useful. And also, as we say, a lot of them were similar across the different classes, so not really helpful for separating them out. The approach used in the paper tended to have more specific features, but had smaller relative frequency of use. So because most of the Go terms were negative, if you're only looking at the positive, they're not going to be used as much. So you can see the rule hits. This is the number of essentially correct times it's identified, are relatively small for some of these. But it did come up with Go terms that seemed to make some sense. So a lot of them were associated with more immunological processes, which, as we know, inflammaging is a thing that is known to happen. So it seems like there may be more biological relevance of the Go terms within the approach that they used compared to the previous approach. So I will go through quickly how we might think about doing this in tidy models. So this is our nice consistent interface that we can use for creating models in R. It makes it easy to move between different modeling packages and lets us try out different model types without having to relearn a load of code. So it's a sort of nice thing to use as we only have to learn one package rather than many for lots of different modeling types. As a quick reminder, the code structure when we're using tidy models is we create a model without any data in it, but we specify the model and the parameters that we want to use. We then create our data, so we do whatever filtering and splitting of training versus testing data or cross-validations. We then combine these two things to train a model, and then we can test and use the model based on the what we trained previously. So how we do this for a random forest, we can use this Ranger engine here. So this is the package that is actually running the model in the background. To call this, we can use this function rand forest to build our model. This has three different model parameters. We have our mtry, which is the number of randomly selected predictors that we saw previously. In the paper, they used 43. We also have trees, which is the number of trees. In the paper, they used 300. And we also have min n, which is the minimum node size. So this comes back to the pruning of our individual trees so they don't get too specific at the end. They didn't mention what this was in the paper, so I've left it as the default in tidy models. The other interesting thing when we're doing this that we haven't seen previously, though, is that we can also add in additional engine parameters. So when we set our engine here to Ranger, we've also added another sort of parameter here, which is called importance. This lets us set a variable importance measure, which is what I have talked about a lot today. So we can set this as impurity, which is our Gini variable importance measure, or permutation for the permutation BIM. The specific approach used in the paper is not specifically available within tidy models, as you might expect, but it does have these sort of main ones here that we can have a look at anyway. In terms of pre-processing requirements for this, there aren't any particular special requirements, although, as I said before, it's worth thinking about any particular biases that there might be within the variable importance measure as to whether we want to use different ones if our predictors are perhaps on different scales or anything like that. So I think I will stick in my continue in my slides for the sake of time. So the nice thing we can do if we have run our model up here and fit it on some kind of model data to predict our class of expression, we can then pull out our important variables. So if we've defined this variable importance measure, we can extract them in a few different ways. So we can visualize them on a graph that looks like this using the VIP package by just passing our particular fit model into this, and we can set a number of features to plot however many numbers of Go terms we so wish at this point. And we can see here that this Go term is obviously quite important and potentially quite a bit more important than any of the others there. We can also pull out the actual values that it's plotted here using this function within the Ranger package of importance. So if we use this function here, extract fit engine, then it will extract the model fit that has been produced specifically by the engine, which means that we can then use this ranger function in a format that it expects based on the original sort of ranger model that was built in the background of tidy models. The final part here, end frame, is just essentially changing the data into a nicer format so that we have a nice tibble that we can look at and sort of filter or whatever in the same way as we are used to in the tidyverse. So that's essentially how you would do that in tidy models. I also, just for the sake of showing a comparison, tried a couple of different VIMs in this model here. Also, just to note, the data that I ran this on was a subset of the data, so these Go terms are not necessarily going to translate to the ones we saw in the paper. I just sort of randomly subsetted it so that it didn't take too long to run. Also, obviously, these importance measures are not the same ones used in the paper either. So for many reasons, this may not correlate. But just to show you the difference between this GVIM versus PVIM, we can see the super important Go term, or the most important Go term, is the same for both of them. Actually, the top two are the same. But after that point, there are some differences. So this 61050 is not as high up, although for the most part they do tend to overlap, but there are then some differences in how important they have been deemed based on the different importance measures. So overall, random forests are nice because they can be quite flexible and they are somewhat interpretable, so you can pull out these interesting Go terms. But we just want to be careful which approach we're taking, as clearly it can alter what we get out at the end, which may have more or less impact depending on the sort of data type that we're using. So that was a look at interpretable models using random forests. This is the penultimate one in this series. So the last one will be on the 21st of August. And we will go through some imaging data in that one and look more at feature extraction for this. So hopefully I will see you all again then. But otherwise, thank you for coming along today.